Thank you very much. So I'm Christoph. I'm working for the Textile Institute of Textile Technology, based in Aachen, so quite near of Maastricht. And we are quite big institute, but never did something with cells. So we joined forces with the DWI, also in Aachen, and started to develop a technology combining textile systems and biotechnology. So at first I think we heard it a lot the last 2 days, why we need cultured meat. Just shortly, briefly, reduce factory farming, the drawbacks we have there, and that's a high potential to get away from these drawbacks. So how we make cultured meat, also briefly, I think most of you know it. Following the classical tissue engineering approach, so extraction of stem cells from animals, isolation of the cells we need, in vitro proliferation, differentiation, and then combination of these short muscle fibers to, for example, burgers. What is the problem we have here? It's restricted to unstructured products. So when you look at the muscle, we have strictly hierarchical structure. So from the muscle fibers bundled in primary muscle bundles, secondary muscle bundles, and the whole muscle structure. And if we want to mimic this structure, we need scalable systems, especially when talking about producing food in the future. So how you can do it in tissue engineering, for example, there are approaches called bottom-up tissue engineering, where you in general take cell-laden structures and assemble them. So we heard it about 2D layers, aggregates, spheres, whatever, or the famous bioprinting. Get a 3D structure, majorate it into the bioreactor, and then hopefully get out a steak in the future. But why are these techniques not suitable now, in my opinion, for cultured meat production? Well, in general, they are limited to small constructs because we don't have a vascular structure. Low processing speeds, talking about scale up, which are difficult to scale up. For example, looking at bioprinting, which is an awesome technology in my opinion, but where you can directly assemble linear structures like we want to have in a muscle. Possible to integrate multiple cell types if we think about fat in the future, and possible to print hollow channels if we want to add, for example, vascular structures. But it's limited by the wall shear stresses. So we can't say we want to print in a velocity of, I don't know, 1,000 kilo per hour or something. It won't work because the cells are just dying in the process. This led me to my idea to use a textile-based technology, namely wet spinning, in this case, where we have a spinning dope which is transported by a pump through a nozzle to coagulation baths, and then drawn to final diameter and collected using a winder. So this also allows us to produce multi-fiber structures. We can also, depending on the nozzle, we have integrate multiple cell types, possible to produce hollow fibers, and reduce, maybe most important part, reduce the wall shear stresses by using large nozzle diameters and then draw the fiber down to our target diameter. So that we at the end get at a higher velocity possible with other technologies, have a fast technology for producing cultured meat. So we started to just produce a few fiber geometries. See, used alginate. We heard in the previous presentation a bit how great this material can be if you use it. And so we produce hollow fibers, bicomponent fibers, multifilaments, monofilaments as well, in fiber diameters reaching from 30 to 1000 micrometer. And production speeds here restricted by the system we used up to 50 meter per minute, with drawing ratios up to 50. We thought, okay, can we use it to like produce meat-like structures? So we colored a bit of alginate, extruded in our spinning system, collected, and assembled it to like a meat-like structure to see if we are on the right way, let's so to say, and then compared the structure with natural meat. Just did a few tensile tests and see that we did a few experiments, and depending on the polymer concentration for sure and the matrix we chose, we're already quite near natural meat. Not completely for sure, but it was just for demonstration. Just to be clear, we don't use any cells here and color this with food color, so it's not a cultivated meat yet. But then we did the first tests to see if we can really extrude cells at high velocities without killing them. So we began to use fibroblasts, extrude them at first just here, call it low condition, just extrude them without pulling any mechanical force onto the fiber into the coagulation bars, and then we enhance the velocity up to over 30 meter per minute, extrude them, and we saw that at first we get a lower fiber diameter by stretching the fibers in the region of 200 micrometer we were at the end, and got really good velocities at this good viabilities, cell viabilities at this stage. So we saw that we can, using a larger nozzle diameter and stretch the cells down, can extrude them at a really high velocity and don't harm the cells. At last step, we used then C12 cells together with alginate and collagen, and used the transglutaminase to cross-link it at the end, and saw that we at first could produce this composite fibers and symmetrical cross-link them, and the Darpion phalloidin staining you see on the right-hand picture shows that cells are growing in the direction of fiber axis, which was quite nice. So our next steps, so first to sum up, it was possible to produce alginate mono and multifilaments, hollow fibers by component fibers at high production speeds, fiber diameters between 30 and 1000 micrometer. We could assemble them in 3D, at least in a cell-free experiment at first, and cells survive spinning speeds at high velocities, higher than 30 meters per minute. As a little outlook, the prospect, we need to find better or more suitable materials for the cell proliferation afterwards. We need bioreactor systems to be able to culture this produced 3D structures, and as a next step, this process has to be examined regarding automatability. So if we want to really use it for high-scale production in the future, we need something to automate the process. Yes, and I'm at the end of my presentation. Thank you very much for listening, and ready for your question.
Thank you very much. Question over there? Oh.
Ambitious.
That's nice, thanks. Hi, thank you for your presentation. I was wondering, how did you sterilize your material before you did the cell seeding?
In alginate, we used UV.
Sorry?
UV light.
UV,
we used collagen, we did ST-well filtration.
And is the UV light sufficient? Because I know when we use UV, it's quite superficial, and the structure is like, we call it like 2D and a half, because it's not 3D yet, but it's also not like in a culture flask.
I wouldn't say it's a perfect sterilization method. Not at all, no.
Then we have some similar issues, I guess. Thank you.
You're welcome.
Hey, thank you very much. There's an online question by Sergio. He's asking whether you have looked through a cross-section of the fiber to see if all cells were aligned, or were they only aligned on the surface?
We used a lot of material combinations. Let's say we had both. So most of them were only on the surface, and a few cases were also in the inner of the fiber.
Thank you.
Thank you. Really interesting work, and very cool to see that the cells can survive those high spin speeds. But my question is for the collagen, did you use any chemistry to functionalize the collagen on the alginate, or was it just simply mixing?
It was just a simple mixing.
Okay, so then the transglutaminase was cross-linking with the collagen then. Okay.
Exactly, yeah.
Thank you.
Oh, yeah.
I'm just curious why it is important that the cells survive. I see this as a maybe downstream process. So yeah.
We aim to really get the muscle out of it at the end. So we want to have our myoblasts, for example, extruded in this fiber, and then they need to survive a process to differentiate in the fiber. So if we
May I
get
ask
dead
an
cells, and
additional question. How do you envision if you have a large construct made of this fiber? How do you envision that the cells remain viable in the inner center?
we have a few ideas here. So one idea would be hollow fibers to extrude in parallel with the normal fibers and then assemble them like in 200 micrometer away from them and then perfuse them. This is well, maybe 2 steps forward, because I think that's the bioreactor could be very complicated, but the other is to use a special bioreactor system to arrange the fibers you produce in a way that the medium can reach them, let's say.
Okay, a final online question.
Thank you very much. Inbar Levav is asking whether, or he first commented that your phalloidine stainings are really beautiful, and he's asking whether you also stained for fast myosin heavy chain to have a more like muscle functional staining.
Not yet. So we are now in this, let's say, screening phase, so that we find a suitable material and then we spend money on antibodies.
Clear answer. Well, thank you very much again.